Hello dear students, welcome to the UGC lecture series on the subject botany. Today our topic is herbarium. In today's lecture, we will be dealing with what basically this herbarium is. Second, we will be talking about its historical background. Then we will discuss the various herbarium techniques, and we will also have a brief mention of digital herbarium. And lastly, we will also highlight the role of herbarium. So let us start from what this herbarium is. A herbarium can be defined as a repository of preserved plant collections, usually in the form of pressed and dried plant specimens, which are mounted on specified sheets, identified and arranged according to a system of classification. The purpose of herbarium is to physically contain the plant collections that serve as a source of research material for taxonomic research and function as reference vouchers for plant identification. If the herbarium specimens are handled with special care, they can remain in good condition for a pretty long period of time. Even they can last for centuries. However, they are susceptible to damage by various types of insects, fungi, fire, water, and therefore should be stored in specialized cabinets. Precautions must be taken against the attacks by fungi, insects, and other such types of external agencies. We have seen the herbaria are typically associated with universities, colleges, research institutes, botanical gardens, natural history museums in different parts of the world. A herbarium may store a few hundred locally collected plant specimens or to the higher extreme of millions of specimens. These herbaria may house flora of a district, region, state, or even broader a country, continent, or sometimes some of the herbaria they house specimens from all over the world. During the last 3 centuries, taxonomists have documented the Earth's plant diversity through these dried reference specimens, which are maintained in herbaria. At present, there are about 3,400 herbaria in the world, with about 10,000 associated curators and biodiversity specialists. Collectively, the world's herbaria contain about 350 million specimens. That is basically the documentation of the Earth's vegetation. In fact, the world's largest herbarium is at the Museum of Natural History in Paris. Now let us have a discussion on its historical background. The credit for the invention of herbarium goes to an Italian by the name of Luca Geni. Since his time, this art of herbarium making was disseminated to other parts of Europe by his students. Herbarium making was a well-known practice even during the time of Linnaeus. Linnaeus is considered to be the father of taxonomy. Linnaeus, in fact, mounted the dried specimens on single sheets of standard size and stored them horizontally, almost similar to the practice what is followed these days. Now let us discuss something about Index Herbariorum, how these herbaria all over the world are indexed. This Index Herbariorum is basically a joint project of 2 different agencies. The first agency is International Association of Plant Taxonomy, and second is New York Botanical Garden. They are together maintaining this Index Herbariorum, and it was started way back in 1935. The first edition of this Index Herbariorum was published in the year 1952. Dr. Holmgreen, a scientist working with the New York Botanical Garden, oversaw the development of Index Herbariorum database, which became available on internet in the year 1997. The Index Herbariorum contains the information about world's herbaria. It lists their names, and the addresses of curators, and also the number of specimens and the type specimens. New herbaria which intend to be registered under the Index Herbariorum must demonstrate that their collection is quite large, usually a minimum of 5,000 specimens, which must be accessible to the scientists, and these specimens must be actively managed. Each herbaria that is listed is assigned a permanent unique identifier code, what we call as acronym, in the form of 1 to 6 alphabets. Now let us discuss the various herbarium techniques. The herbarium techniques, they involve various steps. Out of all these steps, the first step is field collection, that is the collection of plants from the field. It becomes important for a plant collector, a taxonomist, to record certain facts while collecting the specimens from the field. This information must be associated with the plant collections. First, locality, date of collection, habitat type, the associated species, altitude or elevation, collector's name, the local name of the plant, and any other information concerning the plant specimens that will not be evident in the dried and pressed conditions, such as flower color, fragrance, plant size, or even the pollinators. Such information must be associated when collecting a plant specimen from the field. This information may be recorded in a field notebook, or these days we use portable computer systems. If possible, the entire plant, whole plant body should be collected, including the underground parts, in particular in the case of herbs. While as in the case of shrubs and trees, a small twig with leaves, flowers or inflorescence is collected. Along with this field collection, we must have some collection equipments that are must for ideal plant collection. The following items are essential in the field for most of the taxonomic work. These include a field notebook, field press, plastic bags, transparent polythene bags, digging tools, pruning shears, newspapers, a digital camera, and some lenses, especially 10x and 20x hand lenses, a sharp-edged pocket knife, global positioning system, what is called as GPS in the short form, plastic or glass bottles, liquid preservatives, and storage boxes. These are the equipments that must be carried along with collection. One more step in the herbarium making after field collection is pressing or processing of the plant specimens. Once the plant specimen has been collected and field data is recorded, the specimens need to be pressed and dried using a plant press. It is very important to press the plant specimen before it wilts. What is this plant press? A plant press consists of two panels, 1 and 2 panels of either wood or iron, and the size is 12 into 18 inches with 2 press straps or sometimes chains or ropes we use in order to tighten the press. The plant specimens are placed in a folded piece of newspaper. The plant should be arranged in such a manner so that the upper and lower leaf surfaces, flowers, fruits are clearly visible. One more step in herbarium techniques is the drying of plants. Once the plants are in the plant press, the specimens can be dried in several different ways. The press may be placed in the open sunlight or even sometimes indoors. It is very necessary to change these papers at regular intervals until the plants are completely dry. Following drying, the next step comes the pasting, or we call it as mounting of plant specimens. The pressed and dried plant specimens are pasted on the herbarium sheets of standard size. The standard size is 41.5 into 29 centimeters. A paste of glue that is mixed with mercury chloride is used to kill any pest that is associated with the specimens. The adhesive or cellophane tapes are always used to fix the specimens strongly onto the herbarium sheet. Some portions of the specimens, like woody twig, stem, may be stitched with needle and thread. Valuable loose plant pots which can get loose, such as seeds, fruits, they are placed in a small folded packet that is glued or attached with pin on the herbarium sheet. Now the next step comes what we call as labeling. A herbarium label of standard size, the size is 8 into 12 centimeters, is glued, is attached to the sheet, usually in the lower right hand corner, after the specimen has been mounted on the herbarium sheet. In general, the specimen label should include plant's name, family, local name, locality where from we have collected, latitude, longitude, altitude, date of collection, the collection number, brief field note, and the collector's name. After labeling, the next step comes field note writing. The information from the field notebook that we have already noted down when collecting the plant from the field, it is transferred to a computer generated herbarium label or simply a specimen label. The information should be written in black ink so that it should stay for a longer period of time. After this step, the important step is the scientific identification. For scientific identification, the first step is to study the characters of the specimen and check them by comparing its desirable characters with known specimens of the herbarium, or sometimes with various forms of taxonomic literature. After scientific identification, one more step comes what we call as accession. The mounted, labeled and now the scientifically identified specimens are stamped with a rubber stamp or seal, usually on the top of the right hand corner on the sheet. The stamp carries the name of the particular herbarium where the specimen has to be deposited. After accession, we give the accession number. An accession number along with date of accession is added for an accurate record keeping on the accession register as well as on the herbarium sheet. Along with these various steps, we have to also use various chemicals, what we call as herbarium chemicals. As already mentioned, to protect these dried specimens from the attack of insects, pests and other microbes, they are sprayed with fungicides like cresol or sometimes 0.1% solution of mercury chloride. Besides these, other chemicals are used in herbarium such as paradichlorobenzene and naphthalene balls. One more step that we use in herbarium techniques is fumigation. This fumigation involves the exposure of specimens to the volatile substances like mixture of paradichlorobenzene and naphthalene balls, that too in closed chambers for 7 to 8 days. After fumigation, we have to also now fill these specimens into the herbarium cabinets, what is called as filling of specimens. The specimens may be filled in the herbarium according to a particular system of classification in the pigeonholes of the particular herbarium almirah. The specimens belonging to a single species are placed in folders made up of strong paper with light blue color. It is called as species cover. The species covers belonging to a particular genus are often arranged alphabetically and placed inside strong cover of red color called as genus cover. The genus cover of the family are arranged according to the system of classification. Standard herbarium almirahs or cases should be insect proof, dust proof and fire proof with 2 or more pairs of pigeon holes. Now let us discuss the digital herbarium, because this is a recent phenomenon that the herbaria are becoming more in digital format. These digital herbaria are sometimes also called as electronic herbaria. What this digital herbaria is? It is a herbarium in a digitized format. That means it contains a collection of digital images of the preserved plants or the plant parts. The electronic herbaria, sometimes in the short form called as e-herbaria, are often established to improve the availability of specimen data to a wider audience throughout the world. Now let us lastly discuss the role of herbarium. The primary role of a herbarium is to prepare pressed and dried plant specimens, store them in herbarium cabinets and to protect them from the pests. Few more important roles of herbarium are identification of the unknown species. One more role is compilation of biodiversity surveys, as we commonly call them as floras. These herbaria are the source of information about the flora that is growing in a particular region or a locality of a country. Preserved specimens that are in the herbaria, they are used in almost all types of taxonomic research, such as monographic research, revisionary research, or sometimes even the basic floristic work. The diversity and distribution of the world's flora is mainly based on the herbarium specimens. The herbaria preserve the type specimens. These type specimens are the original specimens which are deposited by taxonomists when they describe some new species. Therefore, these herbaria serve as basic repositories for storing the original material collected by the taxonomists while discovering new species. The herbarium material can be used for many different studies. For example, the studies of cytology, palynology, anatomy, phytochemistry, or nowadays molecular biology. Herbarium specimens which are quite old, say 2 centuries old, they have been used for the extraction of DNA. Extraction of DNA has been achieved from these dried pressed herbarium specimens, and then it has led to many great scientific results. More recently, we have seen the herbaria have assumed important conservation role because they are the primary source for information about threatened biodiversity. In particular, the digital herbaria, sometimes also called as e-herbaria, they can be of use for increased public awareness about environment and biodiversity related matters. Some herbaria throughout the world, especially in the developing world, they make their specimen database available on internet so that it becomes easily available in any part of the world. So dear students, today we discussed about herbarium. First of all, we discussed about what herbarium is, then we also tried to trace its historical background, then we talked in detail about the various herbarium techniques. We had also a brief mention about the digital herbarium, and lastly we also highlighted the various roles of herbarium. Hope you have enjoyed today's lecture. With this, I take your leave. Goodbye.